nice pair of beef, a nun's finger bone, and bunions are usually left out of the story of the identification of the remains of the Romanovs, members of the dynasty that ruled Imperial Russia for over 300 years. It's a story that sits on a foundation of mitochondrial DNA analysis, not without some controversy, but it's actually much more complicated than that. The identification of the Romanovs was a major moment in the study of ancient DNA and how we can use it to answer questions about the past. But it's also a story that is still shaping the fields of molecular anthropology and forensic science. This video series will be the deepest dive into the frankly bonkers, but undeniably fascinating story of Romanov DNA that nobody asked for. The Russian Revolution and the fall of the Romanov dynasty is a story that has been told so many times, and I'm not going to really spend a lot of time on the historical background here. There are more in-depth videos and books about the end of the Romanov dynasty. We're gonna speedrun this so we can get to the DNA stuff. The reign of Nicholas II, the last Tsar of Russia, was a disaster, on top of all of the problems Russia already had long before he came to the throne. With the First World War tearing Europe apart and Russia being a total mess, Nicholas abdicated in 1917. He also gave up the throne on behalf of his son, the Tsarevich Alexei. The throne was then offered to Nicholas's brother Michael, but he absolutely did not want that, and he refused, ending Romanov rule of Russia. Nicholas and his immediate family, which consisted of his wife Alexandra and their 5 children, Olga, Tatiana, Maria, Anastasia, and Alexei, were placed under house arrest. Eventually they were taken to the city of Yekatarinberg, where they lived in the Ipatiev house with the 4 servants who had remained with them: the family physician Dr. Eugene Botkin, the valet Alexei Trupp, the maid Anna Demidova, and their cook, Ivan Kharitonov. Early in the morning of July 17, 1918, Nicholas, Alexandra, their 5 children, and the 4 servants were executed by firing squad in the basement of the Ipatiev house. A grave was discovered in the forest approximately 20 miles from Yekaterinburg in 1979 by Julie Ryabov and Alexander Avdonin. However, the idea that this grave could be that of the Romanovs meant that the Soviet Union had to fall before things calmed down in terms of Russia's political situation, and then an excavation took place. That excavation took place in 1991. Archaeologists excavated a shallow grave that contained the remains of 9 adult individuals. This is a bit of a problem, because 11 people died in the basement of the Ipatiev house on that July night in 1918. Nicholas, Alexandra, their 5 children, and 4 servants. What did the skeletal remains tell us about the grave's 9 occupants? The gold, platinum, and porcelain dental work some of them had suggested that these remains belonged to aristocrats. Those sound like some expensive metals and materials to have in your mouth. The skeletons showed a lot of evidence of trauma, including injuries consistent with bullet and bayonet wounds. This trauma does fit with the brutal descriptions of the execution of the Romanovs and their staff. There was also evidence of these individuals' faces having been mutilated and destroyed, possibly in an attempt to prevent their bodies from being identified. Alright, what about the DNA? Buckle up. This is going to be a lot, and we're going to revisit these methods and the results of this first study multiple times. DNA testing in an archaeological context was still a very new thing in the early 1990s. In terms of DNA testing in general, the Human Genome Project, the great big quest to map and sequence the human genome using nice, shiny samples from modern living humans, had only been launched in 1990, a year before the excavation of this grave. Even with this study taking place during the early days of ancient DNA, this study accomplished a lot. So what did various ways of analyzing DNA tell us about these individuals? Let's look at a specific gene first, the amelogenin gene. Amelogenin is a protein that has an important role in the development of tooth enamel, and the amelogenin gene codes for that protein. Mutations in this gene causes X-linked amelogenesis imperfecta, which results in abnormal or even absent enamel, which in turn will lead to other issues with the teeth. Are any of these fun facts relevant to the individuals in this grave? No. It's where this gene lives within the human genome that makes this gene important when it comes to collecting data from unidentified human remains. When we inherit genes, we inherit 2 copies of each gene. You get 1 copy from your biological mother and the other copy from your biological father. These genes live on chromosomes, and you receive 23 chromosomes from each parent for a total of 46. One pair of these chromosomes is referred to as sex chromosomes. because they play a role in the development of the characteristics we associate with being biologically female or biologically male. Most of the population will have either one X and one Y chromosome, male, or two X chromosomes, female. This is because you get your first X chromosome from your mom and your second sex chromosome from your dad, who can give you either X or Y. This is just the basics. It can get a lot more complicated than that, but your copies of the Amelogenin gene live on the X and Y chromosomes. The different copies of the Amelogenin gene, the X copy and the Y copy, are different sizes. When we talk about the size of a gene, we mean how long is the string of base pairs that creates that gene. The X version of the Amelogenin gene is 106 base pairs long, and the Y version is 112 base pairs long. Those bases that make those base pairs are the nucleotides adenine, thymine, guanine, and cytosine, and usually I explain gene construction the other way around. I have a video that explains the basics of DNA in a way that isn't backwards, but the different sizes of the X and Y versions of Amelogenin makes this gene useful for trying to figure out if a skeleton is that of a biologically male or biologically female individual. Alright, let's get back to those individuals in the grave near Yekatarinberg. Looking at the Amelogenin gene of those 9 individuals gave a result of 4 male individuals and 5 female individuals. If we're comparing this to what we would expect if these are the Romanovs, we're missing 1 male and 1 female. If they are the Romanovs, we should expect some of these individuals to be related to each other. How can we use DNA to find those genetic relationships? Short tandem repeats, or STRs, are little chunks of DNA between 1 and 6 base pairs long, and they're repeated multiple times in a person's DNA. There are specific locations for STRs within the human genome, and the number of repeats of these little chunks of DNA varies from person to person. Your particular profile of STRs is inherited from your parents, so you get some of your STRs from mom and some from your dad. Short tandem repeat analysis of these 9 individuals did find a family group, with the skeletons labeled 3, 5, and 6, all female individuals, sharing patterns with skeleton 7, another female individual, and skeleton 4, a male individual, that we would expect to see if skeletons 4 and 7 were the parents of skeletons 3, 5, and 6. So here's what we are looking at with that family group identified in that grave. We'll start with skeleton 7. That is a female individual. We have skeleton 4. And they're a male individual. And so they're sharing these short tandem repeat patterns with 3 other skeletons, 3 other female skeletons in this group. We have skeleton 3. Skeleton 5. And skeleton 6. And again, these 3 individuals are all female. So we can also do mitochondrial DNA and compare these 3 individuals with skeleton 7. If skeleton 7 is their biological mother, they're going to share the same mitochondrial DNA sequences as well as half of their STR patterns. The other 4 adult skeletons, 1, 2, 8, and 9, were excluded as possible parents of skeletons 3, 5, and 6 because they didn't share the right patterns of short tandem repeats. So if these are the Romanovs, we would have Tsar Nicholas II, skeleton 4, his wife Tsarina Alexandra, skeleton 7, 3 of their 4 daughters, 3, 5, and 6, and the family's 4 servants, genetically unrelated to them. We're still missing one of the daughters, and Nicholas and Alexandra's son, Tsarevich Alexei. To further explore kinship and genetic relationships between those individuals and possibly store their names to them, geneticists tested and analyzed their mitochondrial DNA sequences. Mitochondrial DNA is unique and really useful for a number of reasons. All of your DNA lives inside the cells of your body, with the exception of red blood cells, which don't have a nucleus, and egg and sperm cells, which only carry half of your DNA. In the rest of your cells, humans have 2 genomes, the nuclear genome that lives in a cell's nucleus, and the mitochondrial genome, which lives in the mitochondria, the powerhouse of the cell. Your nuclear DNA, also called autosomal DNA, is all of the genetic material you inherited from your biological parents. Mitochondrial DNA, however, is only inherited from your biological mother. That makes it useful for tracing maternal lineages. 6 different mitochondrial DNA sequences existed across these 9 individuals. The sequence of skeleton 7 matched the mitochondrial DNA sequences of skeletons 3, 5, and 6. This is what we would expect if skeleton 7 belonged to Tsarina Alexandra and the other 3 skeletons belonged to her daughters. But this still isn't enough to identify them. To do that with mitochondrial DNA, we need genetic material from someone maternally related to Tsarina Alexandra. Alexandra was born Princess Alex of Hesse-Darmstadt in Germany. Her parents were Grand Duke Ludwig IV of Hesse-Darmstadt and Princess Alice of Great Britain, herself a daughter of Queen Victoria. The mitochondrial DNA lineage shared by Victoria, Alice, Alexandra, and other female descendants of Queen Victoria is sometimes referred to as the Victoria or Queen Victoria lineage. The nice thing about doing these kinds of studies with royalty is the genealogical records that help us find people to whom we can compare DNA samples, because that's what we need when we're trying to use DNA to restore a name to an unidentified individual. Alexandra's mother, Princess Alice, had multiple daughters. One of those daughters, another Victoria, had a daughter, another Alice, who became known as Princess Alice of Greece and Denmark after her marriage. This Alice's son, Prince Philip, Duke of Edinburgh, husband of Britain's Queen Elizabeth II, donated a DNA sample for this study because he would share that same mitochondrial lineage descended from Queen Victoria. Welcome to the Queen Victoria mitochondrial DNA lineage, in which half the people are named either Alice or Victoria. Let's start with Queen Victoria. She had a bunch of daughters, one of whom was Princess Alice. Alice married Ludwig, Grand Duke of Hesse, and they also had multiple daughters. They named their oldest daughter Victoria because that's actually what a lot of Victoria's kids did, to be honest. And then she had a daughter, also Alice. But then Alice had a son, Philip, Prince of Greece and Denmark. He actually had to get rid of his Greek citizenship in order to then become the Duke of Edinburgh and marry the future Queen Elizabeth II of Great Britain. But then we also, of course, have Alexandra. And then she and Nicholas had their 5 children. Olga, Tatiana, Maria, Anastasia, and Alexei. So, all of these people descended from Queen Victoria are going to have the same mitochondrial DNA sequences, the same mitochondrial DNA lineage. His mitochondrial DNA sequence matched the sequences of skeleton 7, the putative Tsarina Alexandra, and skeletons 3, 5, and 6. What about skeleton 4, the putative Tsar Nicholas? We already know that this is a male individual who shares short tandem repeats with skeletons 3, 5, and 6, suggesting a parent-child relationship with them. However, their mitochondrial DNA sequences aren't a useful comparison if skeleton 4 belongs to Tsar Nicholas. We need people who would share his sequences to compare to skeleton 4. So, the geneticists found 2 living relatives who can trace their unbroken maternal lineages back to Nicholas's grandmother, Louise of Hesse-Kassel, Queen of Denmark. This article doesn't provide the names of these living relatives. They might have wanted to remain anonymous, but they are a great-great-grandson and a great-great-great-granddaughter of Queen Louise. The mitochondrial DNA sequences obtained from those 2 living relatives of Tsar Nicholas match the sequences from skeleton 4. Except for one thing. A mutation called heteroplasmy. Heteroplasmy is the presence of more than one type of mitochondrial DNA sequence, called a mitochondrial DNA haplotype, in a cell or an individual. This isn't the same thing as contamination, where you have multiple mitochondrial DNA sequences originating from different people that got into the sample when they're not supposed to be there. Heteroplasmy showed up in multiple DNA extractions from skeleton 4, so the geneticists made sure to replicate that and verify that this mutation really was present in this individual's DNA. So, what does this mutation look like? To understand the heteroplasmy observed in skeleton 4, we have to revisit nucleotides and base pairs. The 4 nucleotides, adenine, thymine, guanine, and cytosine, or A, T, G, and C, are the most basic building blocks of DNA. They're the nucleic acids referenced in the name, deoxyribonucleic acid, and we also call them bases. These bases pair up with each other, giving us base pairs for double-stranded DNA. A pairs with T, and C pairs up with G. Line up a given number of these base pairs together in a specific order, a specific sequence, and you've got a gene. It is more complicated than that, but that's a discussion for another video. The human mitochondrial genome has 16,569 base pairs. Although the paper that told me that is already 10 years old, stuff gets outdated so fast. But the heteroplasmy observed in skeletons for mitochondrial DNA sequences is at one of these nucleotide positions, the place where a nucleotide sits in the DNA sequence. At position 16169, skeleton 4 had 2 nucleotides, cytosine and thymine, instead of only 1 nucleotide. At the time of this study, which was published in 1994, heteroplasmy was thought to be rare, and it's the only difference between the mitochondrial DNA sequences of skeleton 4 and Tsar Nicholas's living relatives. The DNA of Nicholas's mother, Empress Maria Feodorovna of Russia, born Princess Dagmar of Denmark, and his maternal grandmother, Queen Louise, wasn't available at the time of this study to see if they also had heteroplasmy at this nucleotide position, and then maybe that mutation just wasn't passed down to the living relatives. Working with the idea that Tsar Nicholas had heteroplasmy, putting all of this genetic data together led the scientists involved to the conclusion that the remains from this grave belonged to the Romanovs, minus 2 Romanov children, and the maid, cook, valet, and doctor who died with them. Mystery solved, identities restored, right? No, not quite. Not everyone was convinced. The 2 missing Romanov children kept a question alive. Could any of them have escaped? In part 2, we're going to talk about one of the most famous royal mysteries of all time. The DNA drama is just getting started, so thank you for watching, and I hope to see you again for part 2 of the deepest dive into Romanov DNA that no one asked for. We have skeleton 3. I'm gonna spell skeleton.